All right, I want to welcome everyone back. This is going to be screencast number 1 for Chapter 17. And in Chapter 17, what we're going to do is we are going to look at the processes that are involved in taking information that you would find on a gene and using that information to create proteins. So before we get started talking about the different processes involved in creating those proteins, what we need to do is we need to understand the flow of genetic information. Now, the information content that we find in DNA is going to be in the form of specific sequences of nucleotides. The DNA inherited by an organism is going to lead to specific traits, so those characteristics that you would find on an organism, by dictating the synthesis or the making of those proteins. Now, the proteins are going to be the links between the genotype or the genetic makeup of that organism, and then, of course, the phenotype that's going to be produced by the genotype that you find in those genes. Now, gene expression is going to be the process by which DNA is going to direct, again, protein synthesis, and it's going to be found in 2 stages. The first stage is going to be called transcription, and the second is going to be called translation. Now, down here towards the bottom, this diagram is just sort of representing what we would find as we take that information that you find on the DNA and you actually transcribe it, which means to create a messenger RNA strand, and that's what you see down here. And we're going to take the information that you would find on that messenger RNA strand and we're going to translate it. And as we translate it, we're going to create the end product, which is going to be the protein. So what we're going to do is we're going to look at some evidence that was gathered to actually give us some information that verified that genes actually do specify specific proteins within organisms. Now, in 1909, a British physician by the name of Archibald Garrod was the first to suggest that genes actually dictate the phenotypes that you would find in different organisms. And they dictate those phenotypes through specific enzymes that catalyze or encourage specific chemical reactions. So one thing that he had thought about was inherited disease. He had thought that the symptoms of an inherited disease actually reflect an inability in that organism to synthesize or make a certain type of enzyme. So linking genes to enzymes requires the understanding that cells synthesize and degrade molecules in a series of steps, and those steps are very important. And we call those steps a metabolic pathway. So 2 other scientists that we're going to look at go by the name of George Beadle and Edward Tatum. And these 2 scientists, what they did is they developed a hypothesis called the one gene, one enzyme hypothesis. And this hypothesis basically stated that each gene is going to dictate the production of a specific enzyme. And so what they did is they actually created an experiment, and they used bread mold in their experiment, and they subjected this bread mold to X-rays. And when they did that, they actually created 3 different mutant varieties of that bread mold. Now, each variety was unable to survive on what we would consider a minimal medium. So this bread mold has the ability to actually synthesize all of the necessary amino acids that it needs to be able to survive. And so when you put it on a medium that has very minimal nutrients in it, this bread mold can easily survive. But the mutants, on the other hand, they had the inability to synthesize certain molecules or enzymes. And because of that inability, they had a really difficult time surviving on this minimal medium. So using various crosses, what they did is they identified 3 different classes of what we consider arginine deficient mutants. So each lacked a different enzyme that was necessary for synthesizing the end product, which in this case was arginine. So in the diagram that you see on this screen, what you're going to notice is at the very top, this is a very simple representation of the experiment that Beadle and Tatum had carried out. So if you notice in this very first test tube, this is going to be the wild type bread mold. And this is growing in a minimal medium. In fact, both of these test tubes are growing in a minimal medium. But the wild type grows very easily because remember, it can actually synthesize all the necessary molecules or enzymes and proteins that it needs to be able to survive. But the mutants, on the other hand, they could not grow on the medium simply because they were lacking an essential either amino acid or enzyme to be able to eventually create that end product that Beadle and Tatum were looking at, which was that arginine. Now, the results of their experiment are below, and so what you notice is there were 3 different types of mutants that were created. We have class 1, class 2, and class 3. Now, when you look at these three different classes, what you're going to notice is that each class has a specific enzyme that has actually been taken away due to the exposure to the X-rays that Beadle and Tatum had exposed the bread mold to. So if you notice, class 1 mutants cannot produce enzyme A, class 2 cannot produce enzyme B, and class 3 cannot produce enzyme C. Now, each of these different enzymes are responsible for basically proceeding to the next step in the creation of that end product, which if you look down here towards the bottom, is arginine. Now, this is what we would consider that metabolic pathway that we had mentioned in the previous screen. Now, if you look over to the left, what you're going to notice is these were the different environments or the conditions that these different mutants and the wild type were exposed to. Now, the conditions that you actually exposed the wild type bread mold to, it didn't make a difference because remember, the wild type can actually synthesize all of the essential amino acids or molecules that it needs to survive. So if you subjected it to the minimal, it did fine. If you subjected it to where the conditions were you added this particular molecule, which is ornithine, it did fine. Citrulline did great. Of course, arginine, it grew as well. Now, the mutants, on the other hand, remember, they were missing a specific enzyme. So if you look down here, for gene A, that gene A is actually going to be used to code for enzyme A. And so if you take that enzyme out of its environment, it's not going to be able to eventually produce the arginine at the very end. So if you notice, in the minimal medium, it's missing that essential enzyme, so it can't grow. But if you notice here, if you go ahead and add that ornithine, what we have done is we've basically bypassed that enzyme. So once that is present, we can go ahead and complete the pathway and eventually produce the arginine. And then if you look at class 2 mutants, same thing happened. But in this case, for gene B, we got rid of enzyme B. So again, it's not going to grow in the minimal medium because it doesn't have what it needs to be able to survive. Remember, we're missing one of those enzymes. When you add the ornithine, now again, the bread mold is not going to be able to survive because even though you add it, to go on to the next step, which is citrulline, you're missing that enzyme B. But then if you notice, if we add to an environment that has citrulline, then it can go ahead and complete the metabolic pathway, which again is to basically produce our end product, which is arginine. Then of course, class 3 mutants, on the other hand, if you notice, cannot grow in the minimal medium. In other words, they're not producing what they need. Cannot grow in the ornithine, cannot grow in the citrulline, but if you add arginine to their environment, then of course they can go ahead and grow because again, you can sort of bypass those previous steps and you're directly adding that end product, which is that amino acid that it needs to be able to survive. So based on the information that Beadle and Tatum had found, remember their hypothesis was basically one gene, one enzyme. But that wasn't necessarily the case, and you couldn't really go by that because some proteins really aren't enzymes. And so what researchers did is they basically later revised Beadle and Tatum's hypothesis, and they decided to change it to one gene, one protein. Now again, we have to think about this because many proteins are composed of several polypeptides, and each of which has its own gene. So then we had to go ahead and further modify Beadle and Tatum's hypothesis, and so it's now been restated and accepted as the one gene, one polypeptide hypothesis. So you need to note that it is common to refer to gene products as proteins rather than polypeptides. So you're going to notice as we go through and we talk about protein production or synthesis, we're probably going to be using the word protein, but again, it's probably more correct to actually use the word polypeptide. So before we actually start talking about transcription and translation, what we want to do is we want to have a really good understanding of the genetic code. And so what we need to think about is how are the instructions for assembling amino acids into proteins actually encoded within that DNA? So there are actually 20 different amino acids that we find in nature. But you got to think about DNA because there are really only 4 nucleotide bases that are going to be used to code for those 20 amino acids. Now, how many bases correspond to an amino acid? Well, the flow of information from gene to protein is going to be based on a triplet code. In other words, a code of 3. And this is going to be a series of non-overlapping 3 nucleotide words. Now, these triplets are going to be the smallest units of uniform length that can actually code for all 20 of those amino acids. Now, an example that we have here is AGT. So we have adenine, guanine, and thymine. So at a particular position on a DNA strand, this is going to result in the placement of a particular amino acid. and in this case it's going to be serine. And serine is going to be placed at the corresponding position of that growing polypeptide or protein strand. So during transcription, one of the two DNA strands is going to be used as a template strand, and that's going to provide the template for ordering the sequence of nucleotides in that RNA transcript, sometimes called the messenger RNA. So during translation, the messenger RNA-based triplets, and these are going to be called codons, are going to be read in the 5' to 3' direction. So each codon is going to specify the amino acid to be placed again at a corresponding position along that polypeptide. Now codons along a messenger RNA molecule are going to be read by translation in this case, and it's going to be read by this machinery in the 5' to 3' direction. And as we had mentioned earlier, each codon is going to specify the addition of one of 20 amino acids that are found in nature. Now over here on the right, this is just a diagram that illustrates what we had just mentioned. So if you remember, we have a gene on that DNA, so it's going to be a small strand of DNA itself that's going to be used to basically create our messenger RNA strand. So we're going to unwind, we're going to separate this DNA, we're going to use one side as a template, we're going to read it in groups of 3, and again those groups of 3 are going to be called codons, and then eventually we're going to use that group of 3 to establish what amino acid needs to be brought in and connected with other amino acids to eventually produce the polypeptide or that protein. So eventually scientists did crack the code. So all 64 codons were deciphered by the mid-1960s. And so of the 64, 61 code for amino acids, 3 are going to be triplets that are considered stop codons, and actually one of the 61 that you see here is going to be considered the start codon. In other words, it's going to initiate the production of that polypeptide or protein strand. Now the genetic code is considered redundant, and what they mean by that is when you look at these different amino acids, all 20 of them represented on this chart, you're going to notice that each amino acid in most cases is actually coded for by more than one triplet, and so that's what they mean by the redundancy. Now it's not ambiguous, so no codon is going to specify more than one amino acid. Now codons must be read in the correct reading frame or correct groupings in order for that specified polypeptide or protein to actually be produced. Now the genetic code is nearly universal among all living organisms. So what that means is it's shared by the simplest bacteria to the most complex animals. Now this was validated when they actually took genes from one species and transplanted those genes into another, and they were able to be transcribed and translated. So transcription, as we had said, is going to be the very first stage of gene expression, and this can be examined in more detail by looking at the following information. So RNA synthesis is going to be catalyzed or encouraged by a special enzyme called RNA polymerase. And again, we know it's an enzyme because you can see the ASE. And now this enzyme is going to pry the DNA strands apart, and it's going to hook together those RNA nucleotides. Now, RNA synthesis, or the creation of that messenger RNA, is going to follow the same base pairing rules as DNA, except uracil is going to be replacing thymine. So in this case, C is still going to pair with G, but now in DNA, A used to pair with T, but now A actually pairs with U in RNA. Now the DNA sequence where RNA polymerase attaches is going to be called the promoter region. Now in bacteria, the sequence signaling the end of transcription, in other words, finishing up the creation of that messenger RNA, is actually going to be called the terminator end of that molecule. So the stretch of DNA that is actually transcribed is called a transcription unit, again, sometimes called messenger RNA. So the diagram that you see here, this is going to illustrate what we had just mentioned. Now, oftentimes what they'll do is they'll break transcription into 3 different parts. They'll call it initiation, elongation, and termination. So basically what that means is you get the promoter, so this is going to be the area on the DNA where we start transcription. RNA polymerase is going to come in, it's going to initiate the process, it's going to unwind the DNA strand, and we're going to use one side as a template, and we're going to create that RNA transcript or messenger RNA. So this is going to be considered elongation. Now, as we continue with the process, eventually we need to end the process, and so we're going to end up rewinding the DNA. we're going to have the creation again of our final product, the RNA transcript, and if you notice, it's going to terminate by completely binding together that DNA and releasing that completed RNA transcript. So again, in the previous screen, we had mentioned the three different stages: initiation, elongation, and termination. And as we had said, there are going to be those promoters that signal the initiation of the RNA synthesis. Now we do need to talk about something called transcription factors, and these are going to mediate the binding of the RNA polymerase and again the initiation or starting of transcription. Now the completed assembly of transcription factors and RNA polymerase II bound to a promoter, all of this together is called the transcription initiation complex. Now we do need to mention that the promoter is given a special name, and we call it the TATA or TATA box, and this is going to be crucial in forming the initiation complex in eukaryotic cells. Now over here on the right, what you're going to notice is, again, it's just another diagram just sort of illustrating the creation of that RNA transcript. So again, if you notice, this is going to be the promoter and this is going to be the TATA box that we had just mentioned. So it's just a series of T's and A's. And this is going to be the starting point of transcription. This is our template. Again, RNA polymerase is going to come through and it's going to bind here. And these are going to be considered our transcription factors. And down here towards the bottom in step 3, again, the additional transcription factors are going to bind to the DNA along with RNA polymerase II. And this whole thing together is going to be considered the transcription initiation complex. And so that's the inclusion of the RNA polymerase II, again, as we said, the transcription factors, and of course the production of that RNA transcript. So a little bit more information about elongation and termination. So again, as that RNA polymerase moves in, it's going to move along that DNA. Again, as we said, it's going to untwist the double helix. It's going to do it at about 10 to 20 bases at a time. It's going to progress at a rate of about 40 nucleotides per second, and a gene can actually be transcribed simultaneously by several different RNA polymerases. So several RNA polymerases can be working at the same time to get the information from that gene. So termination, on the other hand, again, the finishing up of that RNA transcript is going to be a little bit different depending on whether you're talking about prokaryotes or eukaryotes. So in bacteria, the polymerase is going to stop transcription at the end of the terminator. So it's a really simple process in bacteria or prokaryotes. In eukaryotes, on the other hand, the polymerase is going to continue transcription after the pre-messenger RNA has been actually cut or cleaved from the growing RNA strand, and the polymerase eventually is going to fall off the DNA. Now, even though we're creating this messenger RNA strand, we do need to understand that we might need to modify the strand. And so there are going to be special enzymes in the eukaryotic nucleus that are going to modify what we call the pre-messenger RNA. So this is the initial messenger RNA strand that was created from that DNA template. Now we need to do this before the genetic message is actually taken to the cytoplasm so it can be translated. So during RNA processing, both ends of the primary transcript are usually going to be altered in some way. Usually some interior parts of the molecule also are going to be cut out and the other parts are going to be spliced together. So when we look at modification of the ends, each end of a pre-messenger RNA molecule is going to be modified by the 5' end receiving what we call a 5' cap. And if you look down here towards the bottom, that 5' cap is simply a guanine with 3 phosphates attached. The 3' end, on the other hand, is going to be capped off with a poly A tail. Poly, of course, means many, and what you'll notice again down here in the bottom diagram, we just have a series of adenines that make up the cap of the 3' end. Now, these modifications are going to share several functions. First, they seem to facilitate or encourage the export of the messenger RNA, so in other words, the removal of that messenger RNA from the nucleus of the eukaryotic cell. They do tend to protect the messenger RNA from hydrolytic enzymes, in other words, enzymes that could come through and actually cut up the messenger RNA. And then they also help ribosomes to attach to that 5' end, so eventually we can get the translation of that messenger RNA and the creation of our polypeptide or protein. So as we had said before, in addition to modifying the ends, we also sometimes need to go in and actually make some modifications to the inside of that pre-messenger RNA. Now we do that because there are often what we call non-coding stretches of nucleotides within that pre-messenger RNA. Now what we do is we call these non-coding regions introns. Now, the other regions that actually code for specific amino acids are going to be called exons. Now, these exons are the ones that we actually want to keep. So RNA splicing is going to be used to remove the introns and actually join the exons together. And that's going to create our final messenger RNA strand that's going to be sent out and be used to create our protein. So down here again, you can notice that we have the introns being represented here and here. So these are our non-coding regions. These would be our exons, and so those are going to be the coding regions of our pre-messenger RNA. And so we're going to have enzymes that are going to come through, they're going to take out these introns, then we're going to splice together the remaining coding exons, and again, that's going to produce our final messenger RNA strand. Now, in some cases, RNA splicing is going to be carried out by special molecules called spliceosomes. Now, spliceosomes consist of a variety of proteins and several small nuclear ribonucleoproteins, and these are going to recognize the splice sites. So over here on the right, this is just another example of RNA transcript, again, pre-messenger RNA. If you notice, we have an intron, that non-coding region, so these spliceosomes and maybe some other proteins are going to come in, they're going to recognize this region and basically splice it or take it out. And again, what we need to do towards the end is we need to take those remaining exons and we need to bring them back together or splice them back together. So this is a special molecule that's going to help to encourage this process to happen. Now, ribozymes are going to be another type of catalytic RNA molecule that's actually going to function very similar to enzymes, and it can also be used to splice the RNA. So the discovery of ribozymes actually rendered the idea obsolete, the belief that all biological catalysts really were proteins, because we have another molecule here that can actually do somewhat of the same thing that biological enzymes are able to do as well. So three properties of RNA that enable it to function, as we had said, like an enzyme. The first one being it can form a three-dimensional structure, and again, this is going to be because of its ability to pair with itself. Some bases in RNA contain what we call functional groups. And the third is going to be RNA may hydrogen bond with other nucleic acid molecules. So that's going to help to encourage whatever chemical process needs to happen. So again, remember, that's a function of an enzyme. All right, so that's going to finish up our very first screencast for Chapter 17. And as always, please make sure that you have completed your screencast study guide.